Okay. Good morning, everyone. Can you hear me all right? So I'd like to start by thanking the organizers for the opportunity to come and share some of our work. My name is Kelly Sullivan. I'm an assistant professor at the University of Colorado, and I work at the Crnic Institute for Down syndrome. And we are the largest research home for Down syndrome in the United States. So I'd also like to thank Drs. Jackson, Cook, and Court for giving me a wonderful introduction to what I'm going to talk about today. And where we're going to end up is talking about the safety and efficacy of JAK inhibition in Down syndrome. But where we're going to start is where I started when I came into the field and kind of what my very rudimentary understanding of what Down syndrome was and how we could better understand it. And so, the way that I think about Down syndrome is that it's a collection of different conditions to which people with Down syndrome are differentially predisposed. So if we think about things like Alzheimer's disease, we know that basically everyone with Alzheimer's will have pathology in their brain by the age of about 40, although the onset of dementia can vary by up to 20, 25 years. There's an increased incidence of autism, epilepsy, hematological conditions that we're going to hear more about later. Autoimmune conditions, which Dr. Kaur very nicely introduced, sleep apnea, pulmonary hypertension, the list goes on. On the flip side, the same population is protected from some of the things that are most deadly to people in the United States in any way. So we're talking about coronary artery disease, high blood pressure, and the vast majority of solid malignancies. So that's a really interesting kind of starting point that we've got from epidemiological data. But I think the interesting question, and as a molecular biologist, this is where I come to the table. How do we get from trisomy 21? How do we get from 3 copies of chromosome 21 to this very complicated condition that we understand as Down syndrome, right? If you look again at these different conditions that we know are more or less abundant in the population with Down syndrome, what we see is that none of these numbers are the same. And what that means is we're going to have a different presentation for everyone with Down syndrome. What we also have is a black box here. What is it on chromosome 21 that drives the appearance of these different conditions? We know it can't be one gene. We know there must be some other environmental or epigenetic influences. And I would argue that there are very few of those conditions that we've identified the gene on chromosome 21 that's responsible, with the notable exception, in my opinion, of APP, which we know to be the cause of the predisposition to Alzheimer's disease. And so, thinking of this another way, everyone with trisomy 21 is dealing with it in their own unique personal way. They're all awesome, but they're all very different. And so what this tells us is that we need a personalized medicine approach. One size fits all isn't going to do the trick. We need to treat each individual as an individual, understand their conditions, and then think about what the appropriate treatment regimen is going to be. And so when we ask about what's driving this clinical variation, we can talk about genetic variation. We've heard a little bit about that already. Epigenetic variation, which Dr. Jackson Cook introduced. What's driving these different so-called phenotypes, these clinical presentations? Is it different endotypes? So this is what Dr. Kaur was talking about. Are we talking about differences in gene expression at the level of RNA, as we would measure in the transcriptome? At the next step in gene regulation, which is the proteins or the proteome? Is it in the chemical reactions that are going on in the cells that are represented by the metabolome? What's the impact of environment and lifestyle on these? Is there a role for the microbiome, right? So these are the microbes that live in and on our system and actually vastly outnumber our mammalian cells in everyone in this room. The list goes on and on. There are so many questions. There are so many contributors. And we have a lot of gaps to fill with respect to understanding Down syndrome. So how do we go about tackling a project and a question that is this huge? The way we've chosen to go about it at the Crnic Institute is the development of something called the Human Trisome Project. So this is a cohort study with deep clinical data, a multidimensional biobank, and a public researcher portal. So what does that mean? Well, let's start at the end here with our public researcher portal. So we've actually made all of our data publicly available. You can go right now to trisome.org and link through the Explorer, and you can examine clinical data, metabolomics data, genomic data, immune mapping, similar to what Dr. Kaur was showing, transcriptomics, microbe data, and you can actually interface with the data and look and see what's different with individuals with Down syndrome what's different by age, how do these different molecules interact with one another. It's really just a starting point that we're working with the Include Data Hub to expand, and I'll talk more about this tomorrow. So we launched this in 2016, and in the past 7 years, we've actually been able to enroll over 1,000 participants. In fact, we got participant number 1,000 last week at NDSC. So here you can see our number 1,000 with one of our clinical researchers, Rachel. And one of the things that we've learned is that people with Down syndrome love to participate in research, and we are eternally grateful for that because, as Dr. Kaur mentioned, none of this work is possible without the participation and the support of the community. So how do we generate that data? What we do is we have individuals come in and they agree to work with us, and we start all of this with what we call a digital phenotype. We can't do anything if we don't understand what are the conditions that are currently and have previously affected an individual. So we hand out a participant survey where we ask about a lot of really commonly occurring conditions, and then we ask for full medical records so we can actually kind of cross-tabulate these things, make sure that everything's accurate, and also sometimes we can dig things out of the records that we didn't know to ask when we put together the questionnaire. Then we take a blood draw, and we do a lot, and I'm going to leave it there. And so starting with our digital phenotype, one of the things that we noticed right away that Dr. Kaur, again, has beaten me to the punch on, is that there's widespread autoimmunity in Down syndrome. And this is something that was in the record for decades, but really wasn't looked at from kind of a holistic level to see how prevalent it is and what the potential negative ramifications of it are. And so just data from the Human Trisome Project, we see that more than 60% of individuals with Down syndrome have been diagnosed with at least one autoimmune condition. More than 50% have autoimmune thyroid disease. This is either hyper or hypothyroidism. 25% have been diagnosed with one or more autoimmune skin conditions, 10% celiac disease, and then there are, again, 150 of these conditions, many of which are more prevalent in the population with Down syndrome, although these numbers are much lower, just like they are in the typical population. And you can see, again, they affect many different tissue systems. So another way to look at this is if we look at our individuals, again, from just our study, you can see 75% have at least one autoimmune condition. But once you have one, you're likely to get more than one. And here you can see 23% have two, 9% have three, and 7% have four. And so you can see this kind of cascading effect where we're affecting multiple different tissue systems. Another piece of data that we actually just generated is looking at autoantibodies in adults in the Human Trisome Project. So this is focused only on people 18 plus. So we could kind of capture people after the onset of these conditions that present at different times throughout adolescence and development. And here you can see more than 80% of individuals have either TPO, this is a thyroid autoantibody, or ANA, this is an anti-nuclear antibody. This is a very generic marker of autoimmunity, not specific for any one thing. Or we have 40% here of individuals that have both. And so you can see if we kind of cross all these together, we're getting to the point that we're talking about upwards 80, 90% of people with Down syndrome have evidence for autoimmunity. Okay, so the immune system of people with Down syndrome is dysregulated. So Dr. Kaur mentioned this. We've spent the last decade helping to map this and understand what those differences are. And what that means is that that immune system is mistakenly attacking healthy tissues. So the question here is, what other tissues might be undergoing this attack than the ones that we've already discussed? And what explains this immune dysregulation? Because if we can understand that, maybe we can understand how we can repair that, that damage and actually stop that attack. So one of the things that we have discovered and that we've been working diligently on for the past 10 years or so is the idea that trisomy 21 consistently activates the interferon response. What is the interferon response? You all heard a lot about this during COVID, whether it registered or not. This is your antiviral response. This is one of the first things your body kicks on that says, hey, that's not supposed to be here. Let's mount a response to stop that. And what we've shown, and others at this point, is that people with Down syndrome have a hyperactive interferon response. So again, this is part of your innate immune response. It acts throughout the body. Exacerbated interferon signaling is known to cause autoimmunity. And what we think is that people with Down syndrome are undergoing a sort of systemic sterile inflammation. And just for the record, we have published extensively on this, so I'm happy to talk more about this, but there are publications to go and read. Here we started in very basic, just looking in cells we could grow in a dish. And we found that basically everywhere we looked, there was increased interferon signaling. We looked in the plasma of individuals. We saw signs of autoinflammation. Some of these same markers Dr. Kaur found, elevated IL-6, TNF-alpha, those kinds of things. We showed that there is complete immune remodeling consistent with immune hyperactivity and autoimmunity. We found differences in the metabolome that could affect actually both neurological function and autoimmunity. And just recently, we released our largest data set to date where we kind of define what elevated and hyperactive interferon is doing across different modalities. And we have a lot of work to do, again, to kind of dig through these hundreds of thousands of data points and understand the full depth of it. Suffice it to say, there's a lot there, and we think that we have some pretty good leads to go on. So what does this actually look like? So what we found is that if we take that transcriptome, again, this is the first step. You go from the DNA to RNA. And if we look at all the RNAs in the cell, so we measure about 15,000 of these at a time, depending on the cell type. And we use a piece of software that will help us look for patterns in all of those things, because my brain can't load up 15,000 things at once, but my computer can. We find that we see patterns where if we look in orange, these are things where it looks like that thing's been turned on. And if we look in blue, we look like that thing's been turned off. And what we see here is all of these things in red are related to this antiviral interferon response. And if we look in fibroblasts that kind of sit underneath your skin, we look in lymphoblastoids, we look in monocytes, T cells, these are 3 different immune cell types. We see this orange pattern is the same across all of them, and it's this interferon response. So if we take a number of these genes in order to render something that's less kind of cell type specific and that we can use more generally, what we call an interferon score, we see that these are elevated at baseline in people with Down syndrome. So there's no infection going on. These are healthy people just walking around, and they have more of this going on. And just to compare, this is the same score now looking at typical individuals that have an active COVID infection. And you can see that the magnitude of this elevation is similar when we look at this subset of genes. And so it really does look like, the body is being tricked into thinking that there's something like a viral infection going on when there just isn't. And importantly, this is something that we see that actually doesn't alter across the lifespan. So this is up at a very young age, and it stays up. So this signaling is there very early on, and it persists, which has potentially deleterious impacts. Again, when we look at proteins, inflammatory proteins, IP10, this is related to interferon signaling, TNF, CRP, that IL-6 that Dr. Kaur mentioned, all of these are elevated at baseline, and all of these have been implicated in autoimmunity. Again, when we look at the metabolome, we found a pathway here, this interferon-inducible kynurenine pathway. It doesn't really matter what that is, other than to say it's something that is modulated by this interferon signaling. And a couple of these that are highlighted here, this kynurenine, this is an immunosuppressive molecule that could affect how your immune system's functioning. And this quinolinic acid is actually something that's an NMDA agonist, and it could really affect the way that neurological signaling is happening. And so we can see how, coming from this extra copy of chromosome 21, we can alter signaling that has potentially really broad impacts across systems and throughout development. So where do we think this is coming from? Well, as was mentioned, 4 of these 6 receptors are encoded on chromosome 21. Both of the type 1s, these are expressed on every nucleated cell in your body. So you could imagine having more of these and increasing that signaling could impact basically every tissue system in the body. And then 1 each of the type 2 and type 3. And again, these signal through these Janus kinases, which we're excited about because it turns out these are druggable. And the result is you turn on expression of hundreds of genes in a cell type specific fashion. And those are the guys that we think are actually driving these deleterious effects. And one of the outcomes of this would be this autoimmunity. So why do I think it's these receptors? Other than the fact that they fit very nicely into this picture and we can show that they're there and they're overexpressed. We recently showed in a mouse model that triplication of this locus contributes to a number of hallmarks of Down syndrome, including some non-immune things. So how do we do this experiment? Well, we use something that I assume many people have heard of, genome editing technique that's been in the news for quite a while now called CRISPR-Cas9. This allows us to do really targeted edits into the genomes of various organisms. In this case, we did it into a mouse. Helpfully for us, all 4 of these receptors are in one region in the genome, and there's not any other genes in there that we can identify. So what we did was we cut here, and we cut here, and we deleted those. And we made a mouse that doesn't express those as much as it should. And then what we can do is we can cross this into these mouse models of Down syndrome. So in orange here, we're looking at that locus. So this is a mouse model of Down syndrome where a region of chromosome 16 that has about 120 genes that are also on human chromosome 21 has been triplicated. These mice have a lot of conditions that are consistent with what we see in the population with Down syndrome: congenital heart defects, craniofacial differences, those kinds of things. We can actually take that mouse model and cross it into this one where we deleted it. This allows us to ask, what is the impact of only having 2 copies of those 4 genes instead of the 3 copies that we see everywhere else? It's kind of a subtractive exercise that allows us to say, what is the impact of those extra copies of those genes? And I'm not going to show any more data for this, but I'm going to summarize it. So if we take that mouse model that has a bunch of these different conditions, and we just take those 4 genes from 3 copies back down to 2, we see changes in gene expression across tissues. Again, that's what we would expect based on that model where the endpoint is that we change gene expression. We actually rescue a dysregulated antiviral response that we published a couple of years ago. We rescue congenital heart defects. We rescue developmental delays, cognitive deficits. We even partially rescue craniofacial patterning. So it feels like there's really strong evidence that these receptors are part of driving these things. Not the whole question. There's a lot of other parts to this. I think C-gas is playing a role in some of these, but there's a clear role for the elevation of these receptors in some of the pathology that we see in Down syndrome. So this begs the question, is there a way to normalize this interferon response in people with Down syndrome? The answer is thunder stolen. There are a number of different FDA-approved therapies for blunting the interferon response. This particular class is called JAK inhibitors. Here we can see there's 5 that have received FDA approval, and you can see it's for a wide range of different conditions. Now, this is in the typical population. When we came to this, there was not any evidence of what this medication might do in the population with Down syndrome, which we have to take a very cautious approach because we know that some drugs are not metabolized in exactly the same way in the population with Down syndrome. So we need to be super cautious about assuming that the response we see in the typical population will be the same. Great. So these JAK inhibitors could attenuate many of the effects of interferon receptor triplication. I would also say it's notable based on what Dr. Kaur mentioned about IL-6. These JAKs are used for other cytokine signaling as well. And so one of the beautiful things here is they may have multifaceted use and they may be able to ameliorate things that we haven't really fully contemplated yet. They're taken orally. They have a relatively short half-life, which is a good thing because if there's a bad response, it clears very quickly and we don't have to worry about that being in there forever, which is not true of all therapies. It's fully reversible and again, rapidly cleared from the body. So we already saw part of this picture, but off-label use of tofacitinib and alopecia areata. This young woman actually found me after a conference in 2017 when I first proposed this would be a good idea. And she said, I'm on that medication. And this is what she looked like when I met her, full head of hair. And I said, why are you on that medication? She said, I have alopecia areata, which is autoimmune hair loss. And I said, turn around. Couldn't see any evidence of it, anywhere. She's now been on this medication on and off for about 5 years, and the results are stunning. Here's another individual, again, same, lots of hair loss, 2 months, full regrowth. So we published this a couple of years ago, and this really helped to kind of launch where we were headed with our clinical trial. So the trial we're running right now at the Crnic Institute is looking at 5 different autoimmune skin conditions in one trial: alopecia areata, hidradenitis suppurativa or boils, atopic dermatitis, psoriasis, and vitiligo. And again, these are all autoimmune conditions that are more prevalent in the population with Down syndrome. So our study trial is a phase 2 single-arm open label. That means everybody knows what they're getting and they're all getting the medication. 16-week treatment with tofacitinib, 2-week follow-up. We've actually added a 4-week extension arm because the results have been so great that our participants don't want to stop taking the medication. Adults with Down syndrome, we've actually been able to lower this age. We're down to 12 years old, although the tofacitinib is FDA approved in the typical population down to about 2 years. And again, those 5 skin conditions. AIM-1 safety profile. First and foremost, is this safe? If it's not, we don't want to have anything to do with it. Number two, impact on immune dysregulation. Let's make sure we're doing what we think we're doing. And then aim three, impact on skin conditions. And as a wild swing for the fences, characterization of the impact on cognition and quality of life. It's a complicated study, so we have a large team. Dr. Espinosa, who's the executive director of the Crnic Institute, a number of physicians. We're not MDs, so we need study docs to help us with these things. Study coordinator, a couple of psychologists to help us with our cognitive profiling, and then we generate a copious amount of data that needs to be analyzed and interpreted. This was funded through Include as a phased clinical trial. There's an R61 phase, which is done. I'm going to share just a couple of top-line results. And then we're in the R33 phase now, where we can really grow this out and increase the numbers. So what are those top-level results? First and foremost, 0 serious adverse events. So we've now treated about 13 individuals. We've had no negative reactions that we can directly attribute to the drug. The 6 out of 6 participants with alopecia areata experienced hair growth, although to varying degrees. Atopic dermatitis in 2 individuals saw complete remissions. And then 1 individual with psoriasis saw complete remission. So really, it's doing what we were hoping it was doing, and importantly, safely, and it's working quite well in the population with Down syndrome. But the benefits go beyond skin deep. So when we start to get into the molecular profiling, we're seeing really interesting changes. So again, the vast majority of participants showed a decrease in those interferon scores, which is kind of what we would have hypothesized. We also saw decreases from a number of individuals that had clinically significant antithyroid autoimmunity, and those went down. And so we're getting a lot of different things that we're really excited about. So at n equals 10, there's improvement in cognition as well, spatial memory, visual motor function, and anxiety, depression scores. So just a couple of examples of what this looks like. So again, looking at those transcriptome scores here, again, I showed these data before, but here's control individuals with Down syndrome. These are elevated. This is the median difference that we're looking at here. And here you can see at baseline, our cohort looks like the typical cohort of individuals with Down syndrome, roughly. At 2 weeks, we can already see decreases in these scores, and this persists throughout. This individual here at 16 weeks, that we can see this little blip, this person actually had a cold, and we had to push their final visit by 2 weeks, but then we couldn't push it any further because we needed to get it within the window of when the treatment was going to stop. And so we see this go up, but we think this is indicative of an infection. Relevant to what Dr. Kaur was mentioning, this is really interesting. This person had gotten their COVID vaccine, and that caused their interferon score to spike. And then this went right back down, though, when we looked at the later time point. So again, at the molecular level, the drug's doing what we had hoped. Just another example, because I think these are beautiful and they make me smile every time I see them. Here's an individual, 17 years old, alopecia areata. So this score of 86 means there's no hair on 86% of the scalp. And then when we look at week 16, here's that same young man. And all the hair is back, and he told us he would like to be known as Ed Sheeran. So, and we have happily obliged. So again, looking at what the impact is, you can see for these individuals, we're looking at that SALT score. So now we want things to go down. That means they're doing better. This means the hair is going back. This was actually our first individual, and this young woman had something called alopecia totalis, which is different. It's not patchy hair loss. It's no hair anywhere on the body. But what was interesting is even for this young woman, we started to see eyebrows, eyelashes going back. We think this is something, though, that a much longer course of treatment would be required to see the kind of results that we've seen elsewhere. As I mentioned, those autoantibodies against some of these thyroid proteins, here you can see we had 7 individuals above this clinical threshold, and you can see these are coming down. Now, they haven't completely gone away, but it's a little hint that there are other things that might be improved by this medication. And then just a couple of really tantalizing things. Here we're looking at Cantab spatial span. So basically, this is like the Simon game where we're touching boxes and we're looking at basically how long they can remember that pattern and follow along. And what you can see here is that over time, this is getting better and that they're able to follow further. And then the visual motor test here, we're asking them to trace these patterns, and we're measuring the number of errors that they make in these, and you can see that these are decreasing. So again, these are going in the right direction, and we think we're seeing improvement. Not powered, modest effects. We have a lot of questions about, especially the cognition, but at present, we have every reason to be excited about what these data look like, and where things are headed. So just to summarize, the first phase of the trial was completed successfully despite COVID, which was not trivial. So far, the intervention looks safe. The skin pathology is improved, especially with alopecia, psoriasis, and the atopic dermatitis. We think that there may be benefit for other autoimmune conditions, and we're very keen to go after this. And then there's some tantalizing preliminary results that justify the investigation of potential improvements in neurological function, and Dr. Santoro is going to talk about the SRD later, and we think that there's some interesting potential implications there as well. So acknowledgements, this is going to go way too quick. Obviously, I want to thank all of the individuals with Down syndrome that have participated in our clinical trial and also have given samples and just been overwhelmingly supportive. Funding include project, which is NIAMS, NIAID. We have funding at Crnic from probably 10 different institutes at the moment, all because of the include project. Global Down Syndrome Foundation, who's been a huge backer of ours. And yeah, just everyone on the team. I don't have time to name everyone. And with that, I will take questions.
You guys can speak loud enough. Thank you. What parameters do you recommend for screening? The examples you have shown are in a very progressed clinical state. But in most cases, we have a very low clinical array of symptoms. So what should the GP or the pediatrician do? You do not monitor TPOs very regularly. Maybe you do the TSH, and you certainly do not do ANAs on a very regular basis. How do you find a very early degree of fighting like virus attacks in people with Down syndrome?
Yeah, that's a great question, and I wish I had a more satisfying answer than the one that I'm about to give. All I can say at this point is I think it's too early to think about kind of subclinical and very early things. Everything that we're testing here, which is on a very limited cohort, we need a lot more data, because again, safety is goal number one here, is moderate to severe, right? And the reason we chose the skin conditions is because of what you just mentioned there, right? Some of these things that don't have an outward manifestation are very hard to diagnose. Someone with alopecia, we can spot that very quickly, right? And there's also a very clear readout of whether or not they're getting therapeutic benefit from that medication. So I think that what you're mentioning, these other autoimmune conditions, these things that are presenting less severely, that's the goal, is to get to those things. But I think we just have more work to do before we can even contemplate, what would be the threshold at which we want to start to do these interventions. Right now, it's things that are pretty severe.